The first of what might be many videos going over what we've learned this week in IB Biology 12. This is the fourth week of school. It is the week of September 20th through September 24th. This was an A-day week, meaning we had biology on all Monday, Wednesday, and Friday. And I'm going to try and go over the main topics of what we've learned in class and hopefully explain them in a way that is easy to understand and in bite-sized chunks so that you can either refer to this before we have tests, or if you have questions from class, or if you want to review for the test in several months from now. So looking at this week, what we did on Monday was we went over macromolecules, mainly carbohydrates and lipids, but our homework was to take notes on basically all of the macromolecules, just with the focus in those two. On Wednesday, we talked about the cell membrane, and on Friday, we had an IA day. And so before I jump into this, I'm going to say that I understand a lot of us had some IA struggles today regarding our IAs being changed. And so because of that, I'm going to link a few resources I have found online in the description of this video, assuming I end up posting this on YouTube. And I will also post the link to join the IB Discord server, because I'm pretty sure all of us have Discord, and they have been a great resource for me with IA questions in all my classes. And so if you have questions, they might be able to answer them for you there. So one more thing before we really get into this, I'm going to just talk about the homework that we've had. Obviously, I know you can go on Schoology and look at this yourself, but I'm just going to go over it just in case. So on Monday, she wanted us to do the carbohydrates worksheet, the lipids worksheet, and take notes on cell membranes. On Wednesday, we had a Word document to fill out the questions to, which I don't believe she collected if you made a paper copy. And on Friday, she has assigned us to do notes on a chapter that I have not looked at yet, so I don't know what it's about, and on a PowerPoint that she has posted on Schoology. So try and do those things, I guess. So for the most part, I'm going to go through my notes, and I did little drawings that I'm sure you'll love. Just talking about the concepts. I'm going to try and make this fast, but this week was a lot of content, despite only having 2 days worth of content. So we'll see what happens. So the first thing that I'm going to talk about is macromolecules just in general. So there are 4 types of macromolecules: carbohydrates, proteins, lipids, and nucleic acids. And like I said, this week we focused mostly on carbohydrates and lipids. And there are also 3 kind of like gradually building. So we have the monomers, and, I'll just get in. Why am I even bothering? Monomers, which, let's start with carbohydrates. Down at the bottom here, we see our monosaccharides, which are the monomers for carbohydrates. So these are the 4 that she suggests that we memorize to draw. And yes, I know that sounds like a lot of work, and I'm not disagreeing. You can see I clearly did not draw them here. But this is what she's suggesting we do. And so I put the pictures on screen, and I'd just like to highlight the differences between the 2. So over here, we have alpha glucose, which we see has the OH group here down at the bottom, and the H group up at the top, obviously. And then next to it, we've got the beta glucose, which has the OH group on the top and the hydrogen on its own on the bottom. So if you did not fill out the worksheet that she gave us, it was a packet, and in the middle of the packet had a ton of carbohydrates and their structures. Look at it if you have it. If not, that's okay. These drawings right here basically encapsulate what you need to know. The rest of them you can kind of make up as you go along. And so I'd like to also remind us that we label our carbons 1, 2, 3, 4, 5, 6 is going to be up at the top. So it's going to go around this way. And that's going to become important when we start bonding. So monomers, obviously, we have monosaccharides, and that's what we're looking at here. These are the main monosaccharides. These are not all of the monosaccharides, but these are the main ones. So we have alpha glucose, beta glucose, ribose, and we can see that this is a pentose formation. So instead of having six carbons, it's only going to have 5. So 1, 2, 3, 4, and then again that 5 is going to be up at the top in the CHOH group. And same goes for deoxyribose. And obviously these are only going to have one small, tiny difference, and that's going to be this OH group right here. On ribose, there's going to be 2 OH groups down at the bottom. And on deoxyribose, it's only going to be 1 OH group, and the other is going to be H. So basically for these, the good news is if you can draw 1 alpha glucose and 1 ribose, and you know what to change for the other one, you've got all 4 down. As a reminder as well, IB does not require you to be able to draw and recognize deoxyribose, but she does, and it really is such a small change that it's not that much harder to just learn it. And then the other thing for monosaccharides that I wanted to talk about really quickly is the way that it's written out with the different molecules, I suppose, the atoms that go into it. So C is carbon, H is hydrogen, O is oxygen, and the N is going to refer to the number of carbons. This is all a carbon-based world that we're living in. So if we have, for example, take the glucoses, if we have 6 carbons, it's going to be C6H12O6. Not too difficult, just something to keep in mind. Next, I'm going to briefly touch on the disaccharides, which is 2 monosaccharides. So it's 2 of these one little small guys bonded together. It's 2 of them. So the ones that she has on the paper here that I did not put on the screen, but I'm just going to read off of the paper and you can imagine them in your head, is going to be maltose, which I believe is just going to be carbon. It's going to be a 1,4 bond with carbon, so the number 1 carbon is going to bond with the number 4 carbon, and they're going to line up nice and easy next to each other, so that's just going to be 2 glucoses bonded together. The next one she has on here is sucrose, and sucrose is going to be a bond between a deoxyribose and a alpha glucose, and that's going to be a 1,2 bond, so the number 1 carbon is going to bond with the number 2 carbon. And then the last one she has on here is lactose, which is galactose, which I don't even have on the screen, and glucose. Probably not super important that you know any of these by heart, just something to keep in mind that when we talk about bonding with disaccharides or polysaccharides, which I'll get to in a minute, they are going to bond carbon to carbon, and they're going to be labeled with the number of carbon that we're bonding. And again, that goes clockwise around the carbon, with the last one being at the very top. And so the last thing I'm going to touch on for carbohydrates, and this is the last thing I'll say about carbohydrates, is the polysaccharides. So I'm just going to basically be reading off of the paper for this, but there are 4 different main types of polysaccharides, that we're going to need to memorize. And those are going to be 2 different types of starch, which are going to be used for storage in plants. And the difference is amylose is going to be unbranched, meaning it's just like a spiral, basically. If you can picture a spiral like this, that's what amylose is going to look like. And then amylopectin is going to be slightly branched. So about every 30 of these guys we have, there's going to be a branch like this. So moderately branched, but not a lot. And both of those starches are going to be used as storage for plants exclusively. The next type of polysaccharide we have is glycogen. And glycogen is going to be branched every 8 to 12 glucose. So it's going to be a lot closer together than our amylopectin. And the glycogen is used in animals for storage. It's in the liver specifically. And then the last thing I'm going to touch on is cellulose. And cellulose is used for storage in plants only. Sorry, not storage, structure in plants only. It's hard for animals to digest, but that's about all there is to say about it. She also gave us a paper with matching if that's something you care about. For me, it is not. Moving right along. That was a long bit. Next, we're going to talk about fatty acids or lipids, and I'm going to talk about them very briefly because although they are important, phospholipids is really the main focus of what we need to know the information for. There are basically two different types of fatty acids or lipids, saturated or unsaturated. Now, saturated are going to be in a straight line. So that's going to be like this tail right here. That's a straight line. It's unkinked. It means it's going to have as many hydrogen atoms as possible. So, as many hydrogen atoms as possible, and that allows it to be solid at room temperature. And the other main type of fatty acid is going to be unsaturated, and that's going to be like this guy here. Because there is not enough hydrogen atoms, it is going to form at least one double bond, and that double bond is where we're going to see this kink or this bend that's going to kind of stand out like that. So let's talk exclusively pretty much about phospholipids because I think they're great, kind of. Phospholipids are going to have a head and a tail, as we can see here. They're pretty much always going to be drawn like this or with two straight tails instead of one kinked. But the main thing to know is that the head is going to be polar and is going to be water-philic, meaning water-loving. So that head is always going to turn to where the water is, whereas the tail is going to be non-polar, and that means it is going to be water-phobic, meaning water-fearing. So it's always going to turn away from where the water is. Now, why is this important? Well, because when we drop phospholipids into water, if we only have a few, they're going to stick with their heads in the water and their tails out of the water like they're diving. If we have many, they're going to form a little micelle, which is basically just a circle with their tails all pointing towards the center and their heads on the outside. Or they're going to form what is called a bilayer. And a bilayer is basically just two phospholipids, and they're going to line up like this. So it's going to be two layers of heads with their tails all sandwiched in between, just like that. And the main purpose of why we look at phospholipids in the first place is going to be because of the fluid mosaic model of the cell membrane. So now we are on to Wednesday's content. There's a lot of history on how we know what the model of the membrane is, but the most important thing to know is that we go with the Singer and Nicholson model, and that is the fluid mosaic model like I have written right there. This was proven because we had one human cell and one mouse cell, and we colored one cell, the human cell, let's say, all of the proteins red, and the mouse cell were all green, and then we smushed those cells together and mixed them up, and we were able to see that the proteins mixed together in that cell, meaning that the proteins were not set in stone on where they were in the cell membrane, which is what we had previously thought. There are a few other things that we can look at, but that's really the main reason why we see the Singer and Nicholson model being the preferred model. It just suggests that the cell membrane can change shape. It is not rigid. It is not fixed. It is not a solid protein coating like previously thought. And lipid-soluble molecules can pass through it. So, if we look at the beautiful drawing I have here, we can see that these little circles and the tails are forming a bilayer like we had previously talked about. And there are a few extra little guys in there. So let's talk about them. The one that goes on the outside is going to be a protein called a peripheral protein, which means it is not fully embedded and it is only on one side of the cell membrane. This doesn't mean it can't move, it just means it does not go through it like this one does. This is called an integral protein, meaning it goes all the way through the membrane. These are just an example of some of the proteins that we can see in the cell membrane, and these are kind of the two with weird names, so peripheral, and integral. And then the other thing that we can see I've drawn in here is a cholesterol. These guys are amphiphat. Anyways, they have mainly water-phobic sections, which is like that spiky part I drew, and that's going to align itself with the tails, who are also water-phobic, hydrophobic, but it is going to have one little piece at the very top, which I believe is a hydrogen group. I could be wrong there though, but I believe it's a hydrogen group that is going to align itself with the heads, and it's going to stick out. So what does this cholesterol do for us? Well, it has to do a lot with fluidity. Fluidity, we know that the cell membrane needs to be able to be fluid to pass things in and out, just to do daily things, you know? There are lots of things that can affect fluidity, but here are just a few quick examples. Unsaturated phospholipids, so like we were talking about before with this drawing, this is an unsaturated phospholipid because it has a kink. They're going to be slightly looser because they cannot align as tightly as when they are saturated, like these are. This means that it's going to be tighter and slightly less fluid. In addition, the temperature plays a role in fluidity, and that's because of the cholesterol. In high temperatures, it's going to help keep the phospholipids together instead of just breaking all apart. And in low temperatures, it's going to prevent freezing so that cell membrane remains fluid throughout. And finally, I'm just going to quickly go over some of the attributes of the cell membrane, and those would be that it can flex and break without breaking. It can flex and bend without breaking. So that just means it can bump itself around and it'll be okay. It can repair small breaks, so if there is a small issue with the cell membrane, it can repair itself pretty easily. It can enclose specific organelles in a bilayer. It can create gap junctions for two cells to connect directly and exchange information or materials or whatnot. And finally, in single-celled organisms only, they can do what's called binary fission, where basically the cell cleaves itself in half and creates two new cells. Or two half cells? You get the point. These are going to be the main details about the cell membrane that we are probably going to need to know. If you have any questions with any of these, please feel free to message me and ask. If you have my phone number, you can text me. If you have my Discord, you can DM me. If you have neither of those things, ask in class for one of them. I did not go over all of the material just because I didn't. But if we have questions or anything, or we wanted something explained that I didn't go over, I will obviously do that. But I think that's it for me. I will see y'all on Monday.